continue from the previous class. We started um, we started the tissue system. Is it tissue system? And in that, the internal structure of all now the all organs of the plant we have to learn a little bit. And here is that in the previous class we have learned about the root. Is it a root anatomy we learned? And in the root anatomy there was important point was that the vascular bundles are radial, is it? And protoxylem, that is xylem, is exarch, is it? That is the main point related to the root anatomy. And another thing is that the pericycle that was single layered, is it? This thing you should remember. And also we learned in monocot and dicot. In dicot root, what was the important thing? That it was 2 to 4 xylem and phloem bundles, is it? Xylem and phloem bundles, 2 to 4 in number. And so they were, we can say 2 to 4 means diarch to tetrarch condition. Tetrarch means that di means 2, diarch indicate that when there is 2 xylem patches, and tetrarch means 4 xylem patches, and that is triarch. You can say then there is a monarch, means only 1. So like this way, when it is there, xylem phloem bundles are limited, very limited. This is the usual. Of course, sometime more than this also is found, but usually it is 2 to 4. And there is a pith is in dicot root. The pith is either it is small or absent. Okay, so the pith portion is not very big. And in case of monocot root, this number is increased usually more than 6. Okay, 6 or more than 6. It is equal to 6 or more than 6 generally. And in that case, we call here it is hexarch. 6 is like this way. If we call them, it is hexarch, or we can call the condition is polyarch condition. That is many xylem and phloem bundles are there. And another thing, in case of pith is large. This is prominent. So this way, the monocot and dicot root differ from each other. Now today we have this. Next is that we will learn about the internal structure of stem. Let us see first in the internal structure. First thing that we will see is the internal structure of dicot stem. What is there from the, if we see a transverse section of the dicot stem, then in the dicot stem we will get from the from outside to the inside, we will get in the we will get outermost layer is epidermis. Epidermis is the outermost layer. This one is single layer of cell, and then followed by this is the cortex. And this cortex actually it lie between, this is the tissue which lie between the epidermis and endodermis. This tissue is called cortex, and again this cortex can be differentiated into hypodermis. This is in dicot stem is collenchymatous. Then there is general cortex. It is called general cortex. This is the more bigger portion. This general cortex is parenchyma usually, and then the innermost layer of the cortex, that is the endodermis. So we find here from see this diagram, which is a little bit diagrammatic structure. So here, this is the epidermis. Epidermis is there. This layer is epidermis. Inner to this is hypodermis. It is collenchymatous, and this portion, that green portion, is the, it is cortex, general cortex made of parenchyma cells. All right, and hypodermis. This one is collenchyma, epidermis single layer, and on the epidermis there are, these are the hairs, or it is also known as trichomes. Okay, and also there is a layer of a chemical that is present as cuticle. All right, this cuticle, and this layer, then you can see inner to this parenchymatous cortex, there is this layer is the endodermis, and inner to endodermis is pericycle. Okay, this wavy layer it is showing, and these are the vascular bundles. So you can see the many vascular bundles, they are arranged in ring. They are arranged in one ring or more than one ring. So this ring condition, ring nature arrangement, or the ring arrangement of the vascular bundle is characteristic feature of dicot stick. Okay. This characteristic feature of dicot stem, and at the center, this portion, this portion, this large portion, this is called the pith or the medulla. Okay, so in the center there is pith or medulla, and you can see this portion of the tissue. Okay, that is the tissue in between the vascular bundle. These are coming out like the sun rays, so they are known as medullary rays. So these are the medullary rays. Medulla is the pith and medullary rays are there. Important point related to this anatomy of the stem is there. So vascular bundles. So though many, but there is a limit. Limit is there, and there is the vascular bundles are arranged in a ring, and the vascular bundles are conjoint, collateral, they are open. Open vascular bundles means we have learned about the vascular bundles. Open vascular bundles means cambium, that the meristematic lateral meristem. Cambium is present in between xylem and phloem. So this is vascular bundle is open. Why it is open? Because due to presence of cambium, open for further growth, open for secondary growth. Okay, so the vascular bundles are open. And another thing, in the as it is a stem, so the vascular bundles are, they are endarch. Endarch means protoxylem is xylem is inner. Okay, protoxylem is inner. So there is protoxylem towards the center and metaxylem towards the periphery. That is, so these are the important point here. You can in the description also, here I have written some important point, and the important points are written marked here with a red color. Say this is cortex differentiated into hypodermis, hypodermis, general cortex, and endodermis. Endodermis is also known as a starch sheath because these endodermal cells contain lot of starch grain in it. So sometimes it is called, in the stem, it is called a starch sheath also. Pericycle is multi-layered and sclerenchymatous. In case of root, we have seen that pericycle is single layer. Here in the stem, it may be multi-layered also, and also in multi-layered, it may be heterogeneous or homogeneous. That is made of, when it is made of more than one type of tissue, it is heterogeneous. When it is only one type of tissue, it is homogeneous. So here it is sclerenchyma is there, and alternating with parenchyma. Vascular bundles arranged in, say, the vascular bundles are arranged in ring. Vascular bundles are conjoined, collateral, open with enlarged protoxylem. Central pith is very large and it is parenchymatous, and there are medullary rays. In the monocot, if we see, say, these two diagrams, if you see here, say the vascular bundles are arranged in ring, but here you see in this diagram that vascular bundles, these are the vascular bundles, and many vascular bundles are there of different sizes. Okay, and this different size, they are all scattered from the center near to the periphery. Okay, here also this there is outermost layer is outermost layer is epidermis, and below it there is hypodermis. After that, the ground tissue is continuous from there till the center. Okay, and within that the vascular bundles are embedded. The vascular bundles towards the periphery, their number is more and they are smaller in size. Towards the center, their number is less and larger in size. This is the ground tissue. So the ground tissue system, this ground tissue is not differentiated into ground tissue is not differentiated into not differentiated into cortex and pith. Cortex and pith. Why it is not differentiated? Because in this monocot stem, the endodermis, that the boundary layer, demarcating line, that endodermis and pericycle are absent. So this ground tissue is not differentiated into two portion. So it is ground tissue is continuous below the hypodermis. Okay, hypodermis here, one difference is that the hypodermis here is it was collenchymatous. Here the hypodermis is sclerenchymatous, and due to this, they can provide strength, better mechanical support to the stem of the monocot. All right, vascular bundles. In this case, the vascular bundles are closed. Vascular bundles are closed. That is, the cambium is absent, and as the cambium is absent, so further growth is not possible. So further growth has been closed, checked due to absence of the meristematic tissue cambium, So it is the vascular bundles are called closed. They are like the other stem character. The vascular bundles are conjoined, collateral. Another thing is here the vascular bundles are covered from all the sides, or at least from two sides. It is bounded by a sclerenchymatous sheath. A sclerenchymatous sheath is there. Okay. Sometimes when it is on two sides only, here and here, then it is, we call that sclerenchymatous cap is there. So this provide additional support to the vascular bundles. So this way, this main difference, uh, main characters in the anatomy of, sorry, in the monocot stem, it is differing from the dicot stem, that hypodermis sclerenchymatous. This is a large number of scattered vascular bundles, each surrounded by sclerenchymatous bundle sheath. Okay, vascular bundles are conjoined, collateral, but it is mainly, it is open, and parenchymatous ground tissue is there, no endodermis and pericycle. This is the important point to remember. Now let us come to the anatomy of the leaf. Leaf anatomy, where the vertical section is cut, the leaves of so leaves also monocot leaf, dicot leaf. Here is the part of the section of the leaf, dicot leaf and monocot leaf. This one is dicot, dicot leaf, and here is monocot leaf. Okay, this in the dicot leaf, as every structure, there is three types of tissue system. So here also epidermal tissue system, ground tissue system, and vascular tissue system. So these leaves are bounded by epidermis on the upper surface and on the lower surface. So this is the upper epidermis. This is known as adaxial. Adaxial means it is which is close to the main axis. Okay, here. So it is adaxial, and which is away, that is the abaxial. Adaxial and abaxial. So this one you can also write as a, that upper epidermis, and this one is lower epidermis. Like with the stem also, here also sometime in the epidermis we can get the trichomes. Some of the leaves are having trichomes, others may not have. But there is another one thing that is present, this cuticle. Cuticle on the there, say this pink one is the epidermal layer. Okay, above this pink layer there is a green layer. This green layer is the cuticle. So cuticle is present, And another thing that is on the epidermal system is present is the stomata. All right, this is stomata. Okay, this dicot leaf, they are usually called the dorsiventral leaf. That is, the leaf is differentiated into dorsal and ventral surface. All right, ventral, upper portion, upper surface of the leaf, it is called the ventral, and the lower one is the dorsal. This is back, this is the front of the leaf. So in the most of the stomata have more concentration on lower epidermis. Then come to the ground tissue. See here, this portion from here to here, this is the ground tissue. The ground tissue in leaf, it is known as mesophyll. Say why it is called mesophyll? Phyll is leaf, is it? And meso means in between or inside. That is the tissue within the leaf. So this is mesophyll. And so ground tissue of leaf is mesophyll tissue. And here we can see that this mesophyll tissue, two types of arrangement of the cells. Here you can see the cells are smaller in size, and there are a lot of gaps in between the cells. And just below the upper epidermis, the cells are slightly elongated and they are arranged in row, and it is at right angle to the surface of the epidermis. Is it? So this layer is known as, this is called the palisade tissue. So ground tissue is differentiated into palisade, and this one is like a sponge. As in the sponge, there are a lot of air pores. So here loosely arranged cells are there. So it is called a spongy. A spongy parenchyma and palisade parenchyma. These all the cells, mesophyll cells, they contain very large quantity of the chloroplast containing the chlorophyll. Okay, and the cells are packed on the just below the upper epidermis. This is because they get more sunlight on its upper surface, and so for the photosynthesis they have to trap the light energy, so more concentration of the chlorophyll containing cells just below the upper epidermis. So this is palisade, and on the lower side there is a spongy parenchyma. They have many air spaces. See, the air cavity is there. All right. So they are getting the gases, carbon dioxide, oxygen through the stomata, and these move through these air cavities. So this is, and this one is the vascular bundles. In the leaf of dicot, vascular bundles are many, and the vascular bundles are representing the veins in the leaf. And as in dicot leaf, the venation is reticulate. Okay, so there are uh, you can say the veins are almost scattered, and there is thick veins, thin veins, veins of different thickness are there. So the vascular bundles in the dicot leaves also is large, small, means of different sizes. But each vascular bundle is conjoint, collateral, and in between this you can see the phloem is towards the upper side and xylem below. And the vascular bundle is surrounded by a bundle sheath cells. This is the dicot. Monocot leaf is slightly different from the dicot. In the monocot, what happened? Monocot leaf is also known as isobilateral leaf. Isobilateral, iso means equal, by two, and lateral side means here both the sides are similar in their internal structure. Okay, so they are called isobilateral. And this monocot leaf, as their internal structure is similar, it is actually internal structure of monocot is homogeneous. In dicot it is heterogeneous. So here the this mesophyll cells, they are not differentiated into palisade and spongy parenchyma. So mesophyll is only one type, it is homogeneous. Mesophyll not differentiated into a spongy parenchyma and palisade. And another thing is that the stomata is almost equally distributed on both the epidermis, that is on the upper epidermis and lower epidermis, stomata are almost equally distributed. One special thing is found in the grass leaf. Grass leaf and some plants. In these, what happened here, you can see that some of the epidermal cells, particularly that on the upper epidermis, that is adaxial epidermis. Here, some of the epidermal cells, they are large in size, and it is vacuolated. Say this is epidermis, most of the cells are small and uniform, but some cells are very large and vacuolated. These cells are known as motor cell. Motor cells, or it is also known as bulliform. Bulliform cells. Okay, these bulliform cells, they are very sensitive to water content. So when they have enough moisture, these cells are turgid, fully swollen. Okay, And in that condition, when turgid, the leaf is open, leaf open. But when there is water stress, less water is there, the size of the cell become flaccid. That is, they have less water, and their naturally their volume decreases. And in that case, due to decrease in the size of the bulliform cells, the leaf curls inward, curls inward. So when it curls inward, then the exposed surface is very less. That is, here the leaves roll in. So due to this, the exposed surface of the leaf is decreased, and this reduce transpirational loss. So this is an adaptation against the against that drought condition, that is against water stress. All right. So this is the what is the main difference in dicot and monocot leaf? First difference is in the distribution of stomata. In dicot, it is mostly in the lower surface. In monocot, it is both surface. And after that, the mesophyll. The mesophyll tissue. In dicot, it is there is palisade and a spongy parenchyma, but in monocot, this is not there. And this, of course, this bulliform cells, they are present only in some grasses. Okay, these are the internal structure of the leaf and a stem. So here you can see the monocotyledonous leaf, isobilateral leaf. It is internally difference, they differ from the dicot leaf in having stomata equally distributed, mesophyll not differentiated. Vascular bundles are near similar in size. Say another point that this in monocot leaf, the venation is parallel, and in case of parallel venation, all the veins which run parallel from the leaf base to the leaf tip, they are almost of the similar thickness. So the vascular bundle in the monocot leaf that we get, they are almost of equal size. Okay, next, after this internal structure, we will come to the next thing, that is the secondary growth in plant. secondary growth in plant. What is this? The plant has two type of growth. One is the primary growth. This primary growth is carried out by that the primary meristem, and they are particularly the apical and intercalary meristem. These two meristem, they contribute to the primary growth of the plant, and during this primary growth the plants or plant organ usually grow in their length or height. That is the main growth. Of course it grow in diameter also, but diameter is less, growth in height is more. In some of the plant, particularly that in case of gymnosperm, and dicot, plants, they have this lateral meristem, they have a lateral meristem. Okay, and due to this presence of lateral meristem, there is secondary growth, and in this there is increase in its diameter of organ, diameter of a stem, a root, or other organs takes place, and there is production of secondary tissue. This secondary meristem or lateral meristem, they produce secondary tissue, simple as well as complex. Okay, secondary xylem is produced, secondary phloem is produced, secondary cortex, secondary parenchyma, sclerenchyma tissue is also produced. This secondary growth does not take place in monocot. The secondary growth is absent in monocot plant. In monocot plant, that's why you can see that this bamboo, coconut plant, betel nut tree, no, they grow so tall, they are very big, but their girth in thickness they are very less in compared to the other dicotyledonous and gymnosperm trees. Okay, this is because they don't have the secondary growth. Why they don't have secondary growth, monocot? Because their vascular bundles are closed. They don't have lateral meristem. Okay, their vascular bundles are closed. Of course, there are some uh, plants in monocot where this secondary growth, of course, takes place. Okay, so what happened actually? See, here is this diagrammatic representation of the stem, the different steps in the secondary growth. Here, these are the vascular bundles, so they are arranged in ring. Okay, This is the primary structure of the uh, dicotyledonous stem, where the vascular bundles are arranged in ring. Here, in between the vascular bundles, there is medullary rays, which is a non-vascular tissue, and in each vascular bundle you can see that this blue portion, this is the phloem, this red portion is xylem, and in between this green part, that is the cambium. All right, this cambium, say here written the vascular cambium. Okay, this is vascular cambium is there. This vascular cambium we also call fascicular cambium. Okay, this vascular cambium, the vascular cambium present in between the vascular bundle, this is also known as fascicular cambium. Fascicle is, that is the bundle. So fascicular cambium, and this is as it is present within the vascular bundle, so we can call it intrafascicular cambium. Intra means within, intrafascicular cambium. Okay, during the secondary growth, what happened, there is a formation of cambial ring. There is a continuous cambium is produced. So cambium ring is produced. How the cambium ring is produced? Here the medullary ray cells in the line of the this fascicular cambium, they also become meristematic. Okay, they also become meristematic. So here, this one, see here, interfascicular cambium, in between the vascular bundle, that cambium is formed. So cambium ring is formed by joining of intrafascicular cambium plus interfascicular cambium. This is inter. Inter is in between and intra within. So vascular cambium ring is produced by joining intrafascicular and interfascicular cambium. First thing is that the medullary ray cells become meristematic in that line, and this is interfascicular, so it is becoming continuous, and so this ring is formed. Once this vascular cambial ring is formed, this this is the cambium, then this cambium will produce on the inner side, it will produce secondary xylem, and on the outer side it will produce secondary phloem. So this is secondary phloem is produced, and here secondary xylem is produced. Secondary xylem is produced on the inner side, and secondary phloem is produced on the outer side, and this is cambium. And this way the secondary growth takes place. In places there is formation of some parenchyma cells, and these parenchyma cells, they form the secondary medullary rays. Okay, when this, so this is the ring formation. After that, here it is showing that the secondary growth has started. This blue portion is the secondary xylem. These are the primary vascular bundle, primary xylem. This portion, okay, is showing here. This is the primary xylem portion, and the blue portion is the secondary xylem. All right. And these lines are secondary medullary rays. Secondary rays or secondary medullary rays are formed. So this way it continue. And what happened that the activity of the cambium uh, differ in different time. Ac- Activity of cambium varies with environmental condition. Okay, the environmental condition particularly that is by the fluctuation of the temperature. All right, by fluctuation of temperature. So what happened during the summer or during that the spring season, which is very favorable for the growth and development, during this period the more secondary tissues are produced. More secondary tissues are produced. And during winter, when the temperature is very low or it is autumn, this is not suitable for the growth, and here that very less secondary tissue is produced, and less tissue is produced. At the same time the cells, component cells, are also very small. Here in the summer the more amount is produced and the cells are also very large in size. So the secondary growth which takes place during the summer or the spring season, it is known as that spring wood, or it is also known as early wood, sometimes also called the summer wood. And during the winter, or it is autumn, it is called autumn wood, or it is the late wood. So particularly in the region where the climate is, it is temperate. Actually, in the temperate, there is fluctuation of temperature. In the temperate zone, right? Here you will experience the summer and winter. So in the temperate zone, this spring wood and autumn wood is very prominent. But in the tropical areas where the temperature is almost always high, uh, there is no fluctuation mm, in the secondary growth. So in the temperate region, we get early wood and late wood, or spring wood or autumn wood. So, and every year we are getting these two types of wood, and these two types of wood, they are present in the form of a ring, and this is known as annual ring, or it is called growth ring. Okay. So annually, this is called annual ring because in 1 year, one such ring is produced. This is one such ring is produced. So it is called the annual ring. So every year, one ring is produced, and that's why by counting the annual ring, you can count the age of, you can determine the age of a tree. This, as in case of, say, in the spring wood, slightly lighter in color, okay, and the autumn wood is slightly darker. Their density is high, slightly darker. So these annual rings are very distinct due to this light dark rings, consecutive rings are there. Okay, uh, this is, of course, it is time. Uh, See here in the diagram that here when this secondary growth continues, we will get that this is increasing and increasing. This due to this production of the mm, secondary xylem, they almost crash the primary phloem and the primary xylem. So primary tissues are usually crushed and pressed, and gradually this cambium ring increases and the plant grow in their diameter. Okay, today I cannot complete the whole thing. Some of the other points are still left. So in the next class, I will continue. Okay, do you have any problem? Can you, if you have anything to ask, please, up to here. Any students? Hello? Hello,
No,
can
sir.
you hear me?
Yes, sir, we can hear
There
you.
is no, yeah, yeah, yeah. I have one more thing, that one thing, this is my request, that you can talk to your friends, that, and you can describe, you can explain them about this online classes. Say this now, due to this pandemic, classes are nowhere, that the means f- front classroom classes are not there, but this online classes also have a lot of advantage. No, say first thing, advantages of online classes. First thing, that you are attending, you are learning, right, sitting right in your reading room, no, at your home. Your parents are also not disturbed. Sometimes what happens that your parents have to drop you. A lot of time is lost due to this coming to the institution and going back to your transports, etc., expenditure. Is it? Many things are there. So those things are not there, and anytime sitting at home, you can learn the same thing. This live classes are almost, it is interactive. Okay, this online classes. So you can explain them these things that what the lecture we are delivering. These things are there on the YouTube. It will be uploaded. The link you are getting, you can see the lecture. You can attend the lecture repeatedly. That if you have some problem in understanding, you can repeatedly see these things. So all these advantages are there. Okay. uh, in compared to that classroom classes that usually uh, we all are habituated with. But this also has a lot of advantage. So you can explain, and you can tell your friends also. If they are interested, they can join, because many of the students and guardians, they have this confusion, and they cannot decide and don't know exactly whether this online classes are suitable or it is